on to the next talk, which is by Amit Awasthi. Amit is an associate professor at the THSTI Faridabad. So Amit, you are very well known in the community. Can I please ask you to go a little bit fast so that we can finish by 3:30, so that Shashank will not be angry with me. So please go ahead, Amit.
Thank you, Dipankar. Thank you very much for the kind introduction. And let me share my slide. Can you see my slide now?
Not yet. Yes, now. Yeah.
Yeah, you can see my slide, right? Let me quickly. So basically, yeah, I'll just. Okay, so this is now full screen. So basically, I'm going to quickly touch upon the kind of work what I'm doing from past kind of 3-4 months, trying to understand the immunopathology of SARS-CoV-2 infection in animal models. So I'll quickly just go through. We know that this is a novel coronavirus, which is a respiratory virus and causes disease COVID-19, first identified in the Wuhan. And this COVID-19 really primarily affect respiratory system, like upper tract respiratory system, and further go down into the lower respiratory tract system. So a patient with the severe COVID-19 disease having a kind of very systemic hyperinflammation associated with the cytokine storm. And if you see every other day, there are new reports keep coming from various journals, especially high-end journals, talking about the role of T cells. So basically, the T cells can be also protective in terms of vaccination of against this virus, but as well as the T cells can also cause this immunopathology. So this is kind of dichotomy of the T cell, which is important to understand what kind of T cells can protect and what kind of T cells can cause the disease. So to understand these things, we invested to establish the kind of animal models in THSTI. So this is kind of very quick glance that when the virus is we are infected with the virus, a different cellular system which is in the lung is get activated, and these are the primarily macrophage dendritic cells, T cells, and these cells, once they're activated, induces different kind of cytokines, and these cytokines lead to the kind of immunopathology or disease pathology, you can say, leading to fever and various kind of symptoms. So if you really look into the animal models of SARS-CoV-2, we know this is, I mean, bats are the kind of reservoirs, and they are the one kind of spread the virus, but we don't know how this virus is actually jumped from the bats to the human. That's kind of still kind of controversy, and different schools of thoughts are having different explanation for that. So let's not get into that. So basically, we are kind of more interested to really understand the molecular pathways to understand the disease pathology of the virus. So basically, we have different models which has been shown by different investigators around the world. And I think one of the model which is coming out very nicely is the hamster model, which you can see here. The mice models, normal mice like C57 or BALB/c mice, they are kind of resistant to the coronavirus or SARS-CoV-2 because they are not able to really. There's a kind of the mechanisms which are coming. They are kind of ACE2 is not really fitting that well with the RBD. So there's kind of this interaction which causes this virus to get into the cells is really not as good as human or the hamster. So based on the hamster model is actually showing a kind of good model for studying the preclinical vaccination studies, which is kind of not really that possible even in the ACE2 transgenic mice. So basically, we wanted to understand how this hamster model, what are the different immunopathology and what are the different features in the hamster when we actually infect them with the SARS-CoV-2. So basically, if you look into this kind of very quick cartoon or diagrammatic representation of how the virus is actually get into the cells. So I think 10-15 days ago, we were not really knowing too much about NRP1 and SARS-CoV-2. So initially it was thought that virus get attached to the ACE2, and after this attachment, there's a TRMP-SS2, which is actually cleave the RBD and S protein, and that's how the virus get inside the cell. But a couple of papers get published in Science almost a week or 2 weeks ago, back to back, where they identified the NRP1, neuropilin-1, which is also one of the binding partner for this virus, and one of the peptide of the S1 protein actually get interacted with the NRP1, and that's how this whole process of endocytosis or virus get into the cells. So basically blocking NRP1 is also kind of getting, I mean, people block NRP1 and they found there's a kind of very significant reduction in the viral entry in the cells. So what basically we wanted to understand first, we tried to do this three major protein. So we wanted to understand how these three major protein, one is the TMPRSS2, second one is the ACE2, and third one the NRP1, how they're kind of mimicking, or they're kind of based on the homology modeling, whether they are kind of very similar between the hamster, mouse, and human. So here you can see the blue is the human, cyan is the hamster, and the yellow is the mouse. So this is the so basically the structure for TMPRSS2 was known. So we actually took that CRISPR structure, and then based on that CRISPR structure, we generated the homology models for both mouse and the hamster, and try to superimpose each onto, I mean, these three protein on each other, and you can see they're kind of exactly folded very similarly. So they're kind of very much conserved, and if you can see here, the catalytic site of TMPRSS2 is also kind of pretty much conserved between all these three species. There's kind of some variation you can see here in the yellow with the mouse. So this is the catalytic site for which a lot of inhibitors has been identified in terms of tumor settings. And if you further zoom this catalytic site into the thyroid, which is having two cysteine and one histidine. So this is the three amino acid where this is cleavage happens. So this is also pretty much conserved between the mouse, hamster, and the human. So looks like kind of this is going well in the hamster. And then we went further one more step further to check how ACE2 looks like in the human and the hamster. And you can see these yellow arrows showing there is a kind of, these are the amino acids which are kind of different between the hamster and the human. Again, we did the same thing. The human ACE2 and RBD crystal structure was known, but certain part of this structure was not really fully resolved. So what we have done, we again did the we first modeled that structure, and again did the homology modeling and identify a kind of structure ACE2 with the hamster as well as the RBD, and looks like they're kind of very similarly actually bind with the RBD, both human ACE2 as well as the hamster. And so we focused on the amino acid and the kind of portion which is giving very high value for this. And then we further went into each of these interactions. So you can see each of these interactions between the hamster and the human. We try to identify the interaction energy, and it looks like that there's a kind of very robust interaction between the human RBD as well as the, human ACE2 and the RBD, as well as the hamster ACE2 and RBD. So looks like this is again going well in the hamster model. And after this NRP1 publication came out, we also did this very similar kind of structure. You can see the kind of the blue is the human and the ice blue is the hamster. They're kind of superimposed on each other. And you can see this peptide. This is a S1 portion of the peptide of the virus, which is kind of, if you go further into zooming, you can see there's an interaction which is has been shown in the science paper. We also see that this interaction is actually conserved in the at D320 in the both human as well as the hamster. However, additional interaction we have identified by this modeling that is known and that was not shown in the in that paper. So this is a kind of peptide of the S1 which is interacting with NRP1, and this interaction is the key for the internalization of the virus. If you somehow block this interaction by some small molecule inhibitors or whatever the way, the entry of the virus actually goes down very significantly. So this is also kind of very conserved. So looks like hamster model is kind of not only good for vaccine study, but as well as is good for actually going forward for antiviral screening using these kind of structure, or maybe some different kind of vaccine designing based on these things which can be used in the hamster. So based on this data, we thought it's really good to go into the model and try to identify what are the other similarities which we can identify what happens during the COVID-19 infection in the human. So what we did, we planned an experiment and then we did different time points infection of SARS-CoV-2 in the hamster. You can see we have sacrificed the animals at day 2, day 4, 7, and 14, and we have collected the different tissues, lung, blood, every single, try to collect every single organ and try to understand what are the changes we see in the infection. So kind of hang or okay. So basically you can see what we have done. We have given the 2 different doses of viral infection through the intranasal route, 10 to the power 4 PFU and 10 to the power 5 PFU, and then we measure the body weight of these hamster every single day, and then you can see there's a dip around day 4, which is kind of known in the literature that around 5 to 10% dip normally happens, and then these hamsters actually over time recover. So you can see there's a dip here at day 4, and these hamsters actually losing the weight. And when we sacrifice these hamsters to try to see the gross pathology of the lung at 10 to the power 4 and 10 to the power 5 PFU compared to the uninfected animals at day 2, 4, 7, and 14, and you can see there's a clear sign of pneumonitis and lesions in the lung at higher dose. You can also see at the lower dose, but you can clearly see the lung is very abnormal at higher dose of PFU. And at day 14, these lungs are actually getting better as compared to the early days of infection. And when we try to see what are the other measurable parameters, why which even looking at those parameters rather than going into more molecular aspect, can we say that virus is really these hamsters having infection? And we try to see one of the parameter which was not really shown in the literature is just simple splenomegaly. You can see that at day 2, you can see the spleen size is quite bigger than the uninfected hamster, and it's getting bigger and bigger. And then at day 14, it's actually coming back to the normal. So kind of day 14, there's a kind of recovery in the hamster. So this looks like we can also try to see, and this parameter is very important to record that, splenomegaly is one of the thing which we can also say that there is an infection is high. So then we sacrificed these animals, and then we collected the lung, and then we did the viral PFU. I mean, we did the log 10 copy number per lung mass at 10 to the power 4 PFU and 10 to the power 5 PFU, and you can see at different day. I mean, as days pass away, I mean, days gone. At two days, you have more virus as compared to the 14 days. We also did the viral titer TCID 50 per gram of lung, and you can see at 2 days, very high dose of virus is there in the lung as compared to the 14 days. And we further checked the RBD ELISA, and you can see, and both 10 to the power 4 and 10 to the power 5 PFU, you have a kind of anti-RBD antibody, which is measured here by OD. You can see both of these PFUs giving good antibody response. We further checked other antibodies like spike or NP, and you can see most of these antibodies actually coming at day 14 as compared to early days. So looks like there is a kind of robust antibodies titer you can detect in this hamster model upon intranasal infection. We further went into the pathology of this. So basically, you can see day 0, 2, 4, and 7, and you can clearly see a kind of lung damage in histology. And one of the parameter which also we measure, which was not really reported in the literature, that we try to do a toluidine blue staining, try to see the mast cells, because the idea was that there's a mast cell degranulation, because if you see the virus pathology, it is very similarly mimicking with the pathology of the asthmatic people. So basically, some of the steroid which is actually control the asthma or pathology, that's what we wanted to see, whether there is a kind of mast cell infiltration in the lung, and we could really see there is a mast cell, and these are the different disease score which you can see is increased, inflammation score is increased. So every single thing is increased, and at day 14 these parameters are actually trying to get normal. So looks like this is kind of going well, what we actually seen in previous slides. So I don't know, it's kind of, so basically then we further went into try to see whether there is a kind of some sort of cytokine storm which is reported in the human patient, and we collected this spleen and try to understand what are the different cytokines. So we try to do as many cytokines as possible in the hamster, as these probes were not really known. So we designed our own probes for the cytokines, the chemokines, and the transcription factor for T cells. And you can see at day 2 here, you can see most of the cytokines are elevated, and some of these interesting cytokines which on which my lab is really working from past 5-7 years, like IL-17, IL-9, all these cytokines are kind of very high at early time point, and which is also kind of correlated very well with the TBAT and the GATA3, which are the transcription factor for Th1 and Th2. We also see some of the chemokine molecules are upregulated, and at day 2, which is also kind of a signal that T cells kind of infiltrated, and other monocytes or granulocytes are kind of infiltrated, because this chemokine is really important to bring them to the site of injury. So we did the correlation map of these cytokines and other immune molecules, and you can try to see from this map that if these factors are up, the other factors are equally up. So for example, if the TBAT is up, you can see the CCL4 is up, or PD1 is up. So this kind of, you can put them together and ask the question, okay, what are the cytokines going together to make this cytokine storm, and what are the molecules which are kind of not so together in this cytokine storm? So this is with trying to put the data in a different form, try to say yes, these are kind of all these things are elevated in the disease. So one of the important parameter which we also noticed during the disease is when we isolated the heart, because the reports were coming that something related to cardiovascular, which is also kind of SARS-CoV-2 infection, is causing some sort of issues in the heart. So we try to see the heart, and we can see the kind of gross pathology, uninfected but infected 7 days. This is at the 7 days, and you can see the heart is kind of here more conical compared to the kind of more rounded here. So this is a clear sign that there's a kind of abnormality in the heart, and this normally happens when the walls are kind of thickened, and then heart is actually doing extra work. So as a result, you have this kind of feature in the heart. So then we went into the more detailed analysis into the heart, and then at different time points we try to did this staining, HE staining at 1x, 4x, and then we did the fibrosis empty staining to identify the fibrosis. And you can see there's a clear kind of this interventricular space is kind of reduced at day 7, which is kind of, you can see, is kind of pretty big in early days, but it is kind of reduced. And at day 14, maybe it's getting better. So then we try to see what are the different, whether they are kind of more fibrotic. So we try to see near this artery, there's a kind of more empty stain, and that's kind of sign of kind of fibrosis. We also measure different cholesterol and other TG, HDL, LDL, and one of the thing came out in this is cholesterol is really high in day 4, and also you have very high LDL HDL ratio in these infected hamster. So looks like there's a kind of some sort of cardiovascular issues is also going on in these animals. So we are trying to understand what are the different mechanism for that. So basically, we further went into, and this is still data is under process. We tried to do the serum lipidomics profile of SARS-CoV-2 infected hamster, and at different days, and you can see there is a kind of, compared to uninfected, you have a different days, and different lipids are kind of upregulated. So I mean, we're trying to understand what are these parameters and whether some sort of correlation with this lipid profile with the patient who's having cardiovascular problem. So we are trying to generate a kind of a map which is kind of correlating with the cardiovascular patient. In addition,
Amit, can you round it up soon?
Yes, just this is my last slide.
Okay, great, thanks.
In addition to this, we also did the serum metabolomics, and we have identified a biomarker which is kind of matching pretty much with the serum metabolites of human SARS-CoV-2 infected individuals. So this is the kind of work, most of the work has been done by Jagam, and still is doing a lot of work for various academic and non-academic partners. And Rajdeep and Srikant, who was a PhD student, is also helped initially to standardize this model. We have a team of the people who also helped me in this model. And obviously, this was all started when Dr. Gagandeep Kang was here as a director, and she really wanted this model to be established so that we can really support a vaccine and other academic or non-academic institution in this pandemic time. And thank you very much for providing me this opportunity, and I'm happy to answer any questions. Thank you.
Okay. All right. Very nice talk, Amit, and nice. It's an interesting model. So there are lots of questions. The first one is, apart from heart and lung, are other organs affected? Have you looked into it?
Yes. So we have looked into the liver as well as the intestine, and intestine is actually also having this ACE2 expression. So we clearly see there's kind of we are doing microbiome study, so there is a kind of abnormalities in the intestine.
All right, Amit. So in this model, the hamsters clear the infection with time. Is that right?
Yes,
because they don't die, right?
They don't die. So if you compare ACE2 transgenic mice, they die within 10 days. So you can do very nice survival study. But hamster, they recover by themselves. So there is no mortality associated with this.
Okay. So there was another question regarding the fact that if these mice are if these hamsters are infected, then the problem of transmission in the colony, because I'm sure you're aware that minks in Norway are being culled because they are carriers, right? So are there concerns that these infected hamsters will spread infection in other hamsters in the colony?
Yeah, so so basically that's kind of true, and so there's a transmission study has been done, but what we are actually not really keeping these hamsters once we infect, they are in the ABSL3 isolated facility, and we are not really bringing them back to our regular colony. So yeah, but that's true.
Okay, great. Thank you so much, Amit. Very interesting talk. Thank you.